I think we should start. There's a bunch of people that I haven't met in the room. Hi, everybody. Hola, hola, everybody. Welcome to Phage Friday. Today, we should just do introductions first so that this is like the formal introduction for the recording. So, my name is Adriana Hernandez-Morales. I finished my PhD from the Center for Phage Technology in Texas A&M University. I was the lead scientist that worked in the Patterson Project, and now I am currently at Evergreen teaching a bit of bioinformatics and more Galaxy to the Evergreen Lab. So that's where I'm at. You want to continue, Sabrina?
Sure. I'm Sabrina. I'm the Research Development Director at Taylor. We're a service center at Baylor College of Medicine, and we provide phages for patients, so compassionate use. I'm still going to use that term.
Jean-Paul?
Hello, I'm a phage therapy researcher in the Queen Esther Military Hospital in Brussels, also trying to bring phages to the patient.
And we're also joined by another Taylor member, Justin.
Hi everyone, yep, I'm Justin. I am the lead data scientist and bioinformatician for Taylor. I work with Sabrina here in Houston.
And today we have Michael. Michael, you want to introduce yourself?
Yeah, for sure. Thanks for having me on. Hi everyone, I'm Michael. I'm a second-year master's student in the lab of Dr. Corinne Maurice at McGill University in Canada. And I guess as you can kind of guess by the title, we'll be talking about my work today, focusing on the infant gut microbiome and the phage populations that live there. So I'm very excited to be here.
So for those who don't know, Michael just recently got a paper out on precisely babies and microbiomes, and when do we get phages in our guts, and how do phages get to our guts? So let's start from there. How do phages get to our guts?
Yeah, it's a great question, and I think really only in the past couple of years, or the past I guess 2 or 3 years or so, we're starting to get a better idea of where these first phages are coming from. I think what's generally accepted now in the field is that the first set of phages, which are really detected after about a week or so of life, so pretty early on, come from basically they're induced prophages from pioneering gut bacteria. So I guess the question then becomes, where do these gut bacteria come from? And mostly they're passed down, let's say, from the mother's gut microbiome during birth, or through breastfeeding, or just other environmental sources.
So there's a huge difference there. So it seems that the method of birth is very important for for bacterial colonization, and then that also means what gets to our gut. Have you guys seen any differences between vaginal births and cesarean births in terms of what bacteria colonizes us and what type of bacteria colonizes us, and thus what phage colonizes?
Definitely. So in terms of the bacteria, there are differences between vaginal births and cesarean section births. That's been shown time and time again, but the phage side of things has been not as heavily studied so far. There was a paper that showed, I think they followed children up to a year of age, or it might have been at a year of age, and they showed that indeed, like the phage communities of these children were different depending on birth mode, and also, of course, depending on diet and whether they're formula fed or breastfed, things like that. So I guess same things that would affect the gut bacteria and also affect the gut environment, it seems.
And so what are the first phages that we get in our gut? Is there you guys have looked at the classification, so
yeah. Sorry, go ahead.
No, go ahead. I stepped on you. Sorry.
Okay, no worries. So yeah, the first phages, it's hard to say for sure because a lot of the approaches that have been used are heavily database dependent. And for those of you in the audience who may have been at Evergreen, I guess already 3 or 4 weeks ago, you'll know that not only is phage taxonomy being kind of rewritten from the ground up, but also there are a lot of issues with current approaches and things like that. But if we were to kind of classify things using the taxonomy we have now, what's shown is that at birth there's a lot of caudovirales phages, so a lot of tailed phages, kind of evenly split between the different families there. And then there does seem to be more of a transition to the tailless, the microviridate phages, as the infant ages and matures, and by 3 years of age or so, their gut microbiome in general, so the bacteria and the viruses resemble what we see in adults for the most part. So there's that maturation over 2 or 3 years where the caudovirales kind of decrease and microviridate increase.
And so what about diversity? Is diversity increasing with age and variety of food that we eat? Is it decreasing? What's happening there?
Yeah, so I think diversity is probably a better metric at this stage while taxonomy is being kind of heavily overhauled and we're still left with a lot of dark matter. So in terms of the bacterium diversity, what's been shown is like there's increasing diversity in terms of the gut bacteria that are there between birth and let's say 3 years of age, which is kind of that cutoff where we say it's like an adult-like microbiome. For the viruses, it's a bit less clear, but what's been shown in 1 or 2 papers is that there tends to be somewhat of a drop-off in diversity. So there are a lot of phages that are there at birth, and not all of them can be maintained for whatever reason. Maybe their hosts are missing if they were just acquired from the birthing process, or for whatever reason that they're not able to replicate. And the diversity of the phage community does tend to decrease over the first year and probably into the first 2 or 3 years before stabilizing.
So stabilizing, so this also means that you have I'm taking it to assume that you also have a bacterial population that is in flux. And until that bacterial population is established, you won't have an established phage population. Am I taking this correctly?
Yeah, for sure. I think that's what's probably happening. And because these communities are not only changing in diversity, but also in terms of like abundances, the bacterial and phage abundances early on in life are really low, to the point where like VBRs or MOIs, basically virus to bacteria ratios in the gut, are really quite low at birth, and then they kind of explode within a couple months. At least that's kind of what we see with some data that's been out there. So yeah, the changing bacterial diversity and also the changing absolute abundances of phage and bacteria, I think, definitely have effects on how the community kind of assembles, and then once the bacterium stabilizes, so does the phage community.
There. Sorry, I muted myself. This is very interesting. Is there a driver that we know of in terms of human or pressures from the host, from the human host, to establish this community, like other than maybe diet?
Good question. So, gut bacteria have been implicated in a bunch of human kind of physiological processes. They make vitamins for us. They help digest complex fiber, sugars, help train the immune system. So all sorts of pressures on the bacterial side. But I can't think of anything offhand that's kind of been shown with phages directly with the human host. What we do see, though, is somewhat of a switch from primarily phages undergoing lytic replication early on in life to a more stable and kind of temperate phage or lysogenic replication dominated virome in adults and in older children. So there is that switch there in kind of dominant phage life cycle, which isn't really understood, and that's something we kind of try to create a framework and kind of wrap our minds about and explain maybe in that paper you mentioned. There are different reasons that lysogeny may be preferred in any sort of microbial community, so we highlight those as well.
This is something that makes me think that the fact that there's more lytic phages at the beginning and that there's a switch to lysogeny implies that phages are a big driver of bacterial colonization in our guts and actually bacterial modulation in our guts. And so actually our diet and our access to our environment play a huge role in establishing a microbiome or a quote-unquote healthy microbiome. Am I off?
No, I think you hit the nail on the head there. Diet definitely is important. And like I said, I think of it earlier, different feeding modes and when solid food is introduced have all been shown to affect the virome and the bacteriome. For instance, the kind of ubiquitous or infamous maybe crass phage or crass-like phage group of phages tend to really only appear after solid food is introduced, and that's about the time that these bacteria that digest, or the obligate anaerobe bacteria, which are the crass phage hosts, also appear. So yeah, there's definitely a strong link there with food for sure.
In the paper, you're also proposing like a framework of how you develop your gut virome. You want to talk a little bit about that?
For sure. So like I mentioned, there is this switch that we tend to see in bacteriophage replication strategies, where early on in life there's a lot of lytic replication, which is actually really interesting because we may be able to kind of harness that to our advantage, use that to our advantage, and by therapies to remodel microbial communities at that early life stage before things kind of become too stable to remodel a whole community. But yeah, so we start with this lytic replication, and it does become more of just a lysogenic replication later on, where the community is typically more resilient. So there are different factors that I guess could be kind of applying, or different factors that could be kind of driving this switch. Of course, phages in general can provide a bunch of metabolic benefits to their hosts. So just thinking they can provide different carbon or drug metabolism pathways. Phages also encode virulence factors, so this can also kind of drive that lysogeny forward. They can increase competitiveness, superinfection immunity. So there's a whole bunch of things. I won't go through everything, but phages definitely help their bacterial hosts. But one thing that's been recently shown, and there was this paper by Luke and Silvera in 2020, I believe, so late last year, where they showed that bacterial co-infection by temperate phage promotes lysogeny in the systems they were studying. So that would mean that, for instance, there were several temperate phages infecting the same bacterial cell, or I guess technically more than two. That would be considered a co-infection, and that event there would kind of drive lysogeny. So in the framework we're proposing, we did some kind of back of the napkin math to get a sense of microbial and phage densities and how they're changing as the infant ages, and kind of extrapolating that to the model that the other authors had proposed and published. We do see that there is an increasing tendency for co-infection as the child ages. So really we're proposing that in addition to all the other kind of fitness benefits that prophages can provide their host, that co-infections may actually be the driving factors for lysogeny in the gut, especially as these number of co-infections are increasing with age.
This is super cool. Okay, that brings up a lot of questions, but I also want to open the floor for people to ask more questions, because you already know I talk a lot. So does anybody have questions? And as a reminder, if you don't want to come up and ask your questions, you can send us messages using the little airplane. Sabrina said it was an arrow, but yes, a little
it
airplane.
looks like an arrow.
The paper airplane on the side, and write questions, and we can ask those questions for you. Does anybody have questions?
I guess
Go ahead, Sabrina.
I'll ask. So, what about infants that are more susceptible to sepsis at a young age? Do they have more lysogenic phages in the beginning? Like, is it the opposite? Like, do you see a difference in phage microbiome or virome in diseased infants versus healthy?
Yeah, definitely. So I can't think specifically of any examples of sepsis or kind of enterocolitis, but I know, like, for example, in stunting and in malnutrition, which is quite prevalent on a global scale. I think it's like 1 in every 4 children or so in the world is malnourished at some level. But in these children, we do see like you said, a different viral community that's there, and often it's kind of an immature community. So what we would expect to see, let's say in a child who's 3 years old, we're not seeing that. We're seeing their viral community and also their bacterial community may resemble one that's from a child that's, let's say, 1 year old, 1 year of age. So there is that kind of microbial immaturity in these children that's associated with disease.
So does that mean that impacts them for life, like long term?
Yeah, I think that's the big unknown at this point. I'm not sure if there have been any longitudinal studies like that far to study beyond just childhood. But that would be really interesting to see, like, do these effects kind of sustain themselves over 10, 20 years, and what effects they have then.
This is very interesting. Because the other thing that I was going to ask you about, and like this to me is very interesting to determine, is how do you determine phage host interactions? So phage bacterial host interactions in the gut. How do you know that which phages infect what, and what kind of co-infections are actually happening?
Yeah, and I think that's the golden question at this point. It's quite difficult because a lot of gut bacteria aren't easily culturable. And then beyond that, it's a matter of isolating phage, which is like a whole extra step. So once you culture the host, you of course need to isolate the phage, which if they're primarily temperate, that may not be too easy. But there are some newer methods that have been kind of described and published in a couple of papers to do so at a community level. Of course, there's the in silico approach of CRISPR spacer matching. So that's basically a matter of sequencing your full communities and matching your phage genomes to CRISPR spacers that you're detecting in the bacterial genomes. And of course, CRISPR is the bacterial immune system. So if you find a match, it's probably a good sign, a good hint that this is phage host pairing. Beyond the in silico approaches, though, there are kind of more wet lab approaches, if you want to call it that. So viral tagging is one that we talk about, and I'm in the process of kind of getting up and running in our lab. Briefly, it's a matter of kind of incubating fluorescently stained virions or viral particles from your fecal sample with the bacterial community from that same sample, and on a flow cytometer, you'd be able to detect 2 separate populations where bacteria that have a phage attached to them, which you can kind of infer is perhaps a phage host pairing, would fluoresce brighter than bacteria that don't have any phage attached to them. And you can purify the population and work with that as you wish. What else? There's always a bunch of other approaches and new ones coming up every other week, but maybe another cool one I'll mention, and we also wrote about, is IC or chromosome conformation capture, I think, which takes advantage. And I think there was the talk at Evergreen from the group at Phage Genomics. They spoke about basically how their method and what they've kind of got up and running with phage host pairings in the gut takes advantage of the fact that phage DNA is inside bacterial host cell during infection, so they can ligate. I mean, I don't know all the technical details, but it's essentially a matter of fixing your sample and then ligating the phage and bacterial DNA that's in the same cell, and then sequencing that to get some chimeric reads that give you a sense of which phage was in which cell, kind of thing. So, yeah, there are a bunch of approaches, and thankfully there are also more coming out quite often.
That is super. This is super cool. So this goes to a segue of we should talk more about methods, because this is super cool and they're new methods.
Yeah, methods are great. Love hearing about new methods.
There were a couple of questions based on phages and establishment of phage/bacterial populations. And so the first thing is that you just mentioned that lytic interactions are going to dominate at the beginning, while your bacterial population gets established. And once that happens, then it's going to shift to more of prophage induction, and then thus a lot of lysogenic phage are going to dominate in the population. How variable is that during infancy? And the reason why I'm asking is there are a couple of association studies that have been published already where different types of bacterial families have been associated with disease on the long term in humans. And you already mentioned that there are not a lot of longitudinal studies, but in the literature, in terms of establishing the microbiome during infancy, how much can that microbiome change, and until when? If that makes sense.
Yeah, no, that makes perfect sense. That's something we're actually looking into now to see, like, at what point does this, I call it a switch, or maybe you can call it like kind of a change in life cycle. When does that really occur? Is it variable depending on the infant, or kind of what factors help explain that? And I think really determining that transition time point is pretty key, so that we can kind of target therapies better, if that makes sense. So ideally we'd want to target any sort of, let's say, microbiome remodeling therapies during this kind of early time point where we know lytic interactions dominate. We don't have to worry too much about like a resilient lysogenic or resilient temperate community. And yeah, I guess that's still kind of up in the air, but I would say it is somewhere during the first 3 years of life, because that is when we see the switch at 3 years. It's already lysogenic replication mostly. And yes, there is definitely a need for more longitudinal studies studying both the virus and the bacterium, seeing how they co-evolve and their co-evolutionary dynamics, which is also really understudied.
Go ahead, Jean-Paul.
Hi, Michael. First of all, I need to apologize. I didn't find the time to read your paper, but I wonder whether you looked at the impact maternal infections and or broad-spectrum antibiotics on the composition and diversity of the neonatal microbiome and virome.
Hi, Jean-Paul. Yeah, great question. So I haven't looked at that directly myself as of yet, but there have been studies out there that show that maternal consumption of antibiotics, especially during the later part of pregnancy and right around the time of birth, can alter the infant's bacteriome, which I mean, then we can kind of extrapolate and say it probably alters the virome as well, because they typically go pretty hand in hand. And that could be due to a bunch of reasons, like maybe some antibiotics do make their way into the breast milk or into the infant one way or another. But also there's evidence for vertical transmission of phages and bacteria from mother to child. So if there are any alterations in the mother, we would also expect to see these in the child as well.
Thank you.
Great question, thanks.
Are there any other questions or any questions from the audience? Because I just asking a bunch of questions as always. Hey, everybody. Are there any questions from the audience? Please raise your hand or send questions via back channel. We could ask those questions for you. Are there any questions? I'm sorry. Did I miss? Okay. So then I do have more questions. So what about replication cycles of phages within the gut? Is there anything known about that?
So would you, are you referring to like
do phages produce the same amount of progeny, or is there a way for people to predict how well phages are replicating and how many phages are replicating in the gut? Is that
Well, I think,
possible?
I don't recall ever reading or seeing anything about that. That is something we're kind of working on the side now, but it's still in its early stages, so I won't talk too much about it. But it is hard to tell without actually doing, let's say, the full culturing, let's say, and isolating phages and hosts to see what birth size is and things like that. Of course, there are always like crude methods by, let's say, studying phage and bacterial abundances. So what we do in the gut quite often, we can get these nice, gorgeous plaques that most phage biologists get, just because we have so many hosts and so many phages. So we do counts either on the flow cytometer of our phage particles or through efflorescence microscopy. So just staining phages with any sort of stain and then enumerating them that way. But it is difficult to get a sense of like birth size and in general like tracking phage abundances. There's always, of course, sequencing as well, which you can use to track kind of relative abundances, since there are different biases in the sequencing pipeline. So it's quite crude, I guess, at this point.
So that was the segue to the next question, which is, are there or have there been any studies determining phages and like phage pharmacodynamics in the gut? Because there are a bunch of products now that are out there that want to modulate phages in the gut, whether it is because you get traveler's diarrhea, or because you get or because kids get sick, or because you want to change your microbiome and this is going to be the new therapy. How does that affect phages in the gut? So if we don't know yet how many phages are reproducing and how much phages are reproducing, is there a way to determine how the impact of those treatments later in life or earlier in life as well?
It's a good question. So at Evergreen, actually, one of the talks was by Dr. Tegan Brown at the Quadram Institute in the lab of Dr. Evelyn Adrianson's, and she was looking at the effects of these kind of phage cocktails, I guess, like, for example, the intestibacteriophage from the Eliava Institute. So the effect of these different cocktails on, let's say, just healthy individuals. And I think in most of her kind of the individuals that she studied, she didn't see any significant changes, which is good, which means that these effects of the phage are quite specific to the pathogenic hosts, which weren't present in these healthy individuals. So in that sense, there's no off-target or unforeseen effects of these phage therapies in the gut. But I think in terms of like whole virome transplants, or I guess transplants would be the right word, there has been, there was a study in, I think it was 2018, published in the journal Gastroenterology, and essentially they looked at phage or whole virome transplants in the gut for treating Clostridium difficile infection. So this is a bacterial infection of the gut which causes severe diarrheal disease in many individuals around the world. I don't know the number offhand, but briefly they found that these phage I call them phage transplants, but they called it, I think, a filtrate transplant because it was anything that was smaller than bacteria. But yeah, these filtrate transplants had basically or almost just as good an effect as just the conventional therapy, which is a fecal microbiota transplant. So it seems that even in adults, at least for treating certain types of pathogens, this kind of whole virome transplant or even intestibacteriophage, some sort of oral cocktail, is effective when targeting specific kind of pathogens. In terms of the effects on remodeling the whole bacterial community, that's a bit more variable and I think needs some more study. There's some work being done in mice now, some whole virome transplant studies that are being done. So it'd be interesting to see what comes out there.
My guess is then that these cocktails would have a larger impact in the infant and baby gut, since it's a community that's being established. Oh, this is a question for Jean-Paul, and maybe Maya, who's down there. How do kids receive these phage cocktails in Georgia and in places where intestiphage is being used, for example? Do you know, Jean-Paul?
Well, I think Maya is in Georgia. This is a question for Maya, I think.
Maya, can you come up? I'm going to invite you up and see if invite to speak. I wanted to know. Yes. Hey, Maya.
Hi, everybody. So the question was how they receive orally. It's mostly received oral. You mean administration route?
Yes, but I'm asking if kids get the same cocktails, are they administered these cocktails?
Yes. Yeah. It's the same. Just the dosage is different, but they can be administered orally and also rectally, if it's a correct term.
Yeah, it is. So,
but dosage is different. Of course, it's smaller doses are received by children, and it also depends on the children age. Below 5 and after 5, between 5 and 12 years old. It's different dosages. Yeah, but the cocktail is the same. Yeah.
Thank you, Maya. So this gets me to think that these cocktails have maybe a larger impact in the infant gut, based on what Michael's just saying, that since the bacterial population is being established up until about 2-3 years of age. Am I correct, Michael?
Yeah, somewhere around there.
Yeah, so in children under 3, then maybe these cocktails have a larger impact in what actually gets colonized, what bacteria colonize the gut and what bacteria don't colonize the gut.
I wonder if you compare it to antibiotics, would it Would it be?
That was my next question. What about comparing the effect of these cocktails in infant and versus what happens with antibiotics and giving a kid an antibiotic?
Yeah. Go ahead, Maya.
No, you go on. Yeah, I will let
I'm just going to say I imagine the effects of antibiotics obviously are a lot broader generally. But yeah, I'd wonder to see, I'd be curious to see, like, what the effects of these phage cocktails are in these really young children. I guess at the end of the day, it may depend if their hosts are there or not. But if they are, then it may have, like you said, a bigger impact in these children.
Yes, of course. The antibiotics will destroy all the microflora there, and phages will have more specific activity. Also depends what kind of problem is the child has happened, against which pathogen the child is treated. So it can be different, very different from each other.
Yeah. So if I'm not mistaken, most of the cases of infant that need to be treated with antibiotics, not necessarily. So in terms of gut disease in kids, are related to E. coli, diarrhea, and dysbiosis. Am I wrong? You were going to say something, and I interrupted you. Sorry.
No, it's okay. I had a different question, so I think it's better to continue with the thread. I think.
Okay. Yeah. So
I think there are some low birth weight babies that are given antibiotics prophylactically. So not just those with the disease.
Yeah. No. I mean, those cases might be different, but I'm wondering if you give. So my take on this, and I don't know how far away I'm from like what is actually happening. My take on this is if kids receive antibiotics before the age of 3, that is actually going to delay the establishment of a healthy gut microbiome. And so what Michael is talking about, looking at these establishment of these communities, and then you're still going to see a lot of lytic phages dominate in the population over time until maybe a longer period of time, just because you just disrupted everything with an antibiotic. Am I that wrong in this prediction?
Sounds like a good hypothesis, yeah.
I don't know if this has been seen before, but then
antibiotics in infants, especially the earlier on, I think it's been shown to have like these longer term effects that you're mentioning. So, I wonder if it does delay the progression that we're seeing in the phage community.
I kind of had a question related to that, more like a question about a general question about the virulence. How stable is virulence once it gets established? Is it susceptible to disruptions by things other than antibiotics? Diarrhea is a pretty big change to the gut microenvironment. So does traveler's diarrhea cause significant widespread changes outside of E. coli phages? Again, I'm not familiar with the literature, so I'm not sure if this has been looked at.
Yeah, sorry, I just missed that. You're asking how stable the phage community is in all these different diarrheal diseases. Okay. I guess like same way the bacterial communities kind of shift quite a bit during these diseases and often for quite a while after. I imagine the phage community would also shift. I know, for instance, in IBD, so inflammatory bowel disease, that's an inflammatory disease of the colon and really the whole GI tract. There are changes in the gut phage population during when patients are healthy or when they're in an active disease kind of flare stage. There is definitely a pendulum motion between different states depending on how the host is.
Thanks.
Go ahead, Jean-Paul.
Oh yeah. So I read also some papers that found that the early life microbiome is also important for the development of the innate and adaptive immunity. And if you would disrupt this early microbiome with antibiotics, this would of course also have an impact later on and could cause disease like asthma, for instance. But I had another question. These fecal microbiota transplantations, when you see how they are performed, the fecal suspension is made hours before transplantation using a blender, for instance, and knowing that most bacteria in the microbiome are anaerobic or microaerobic. This would mean that what is transplanted in fecal transplantation is also mostly phages or metabolites. But my question is, there are indications that the phage transplants, they work, but do you think that it would be possible to target, for instance, Clostridium difficile with a few phages, or would this just be absorbed by the microbiome?
Good question. Yeah, I guess ideally, right, we would have a few phages to target any sort of pathogen in the gut or any other part of the body. But like you said, the immune system is quite involved, especially in the gut. It's a very rich environment, a lot of immune cells in the intestinal epithelium there. And phages, there was just I think it was a Nature Medicine paper a few weeks ago from Graham Hatfield's group where they showed that phage immune interactions are perhaps even stronger than we thought, with antibodies sequestering phages completely in one therapy patient. So I think we have to be kind of mindful of how we use phages in any part of the body, and ideally, like you said, using cocktails, when possible, with a couple phages. But yeah, if we could use, for instance, if we could eventually purify or identify certain key phages that are responsible for targeting C. difficile, we don't have to do a whole virome or a whole microbiome transplant. I think that would be much more efficient and efficient use of our resources.
Thank you. And you mentioned this Nature Medicine paper, and for us, but I'm also sure for Maya and for Thea from the Eliav Institute, we were a bit surprised because I think since the 30s there were papers saying that you get this antibody reaction, this immune reaction. I think in a matter of 1 to 2 weeks to phages. I think it's even in some inserts of phage products in Russia. It's mentioned do not use them longer than 2 weeks. So I was a bit surprised by that paper.
Yeah, as was I. It was interesting to see, and also, I mean, considering how we hear stories all the time, and we read other papers that patients are treated with phage for longer than 2 or 3 weeks without any issue. So I guess there might be more profiling that needs to be done in terms of immune status or perhaps some mild kind of immunosuppression. It's a bit beyond me, so I can't comment too much on that. But it's interesting to see though.
Peter Yan, you come up here. Hi. You have a question? I don't know if I can't hear you if you are unmuted.
Hello, can you hear me?
There you are. Yes.
All right. Hello, everybody. I have a question. I have to join. Michael, this is very, very interesting. As Jean-Paul, I did not have time to read your paper, so this might be a stupid question, but nevertheless, I think the shift, what you say, from the lytic to the lysogenic phage population, It's quite a general statement, so I was just wondering about sample biases, and I was wondering actually what about the types of samples you analyzed. Was it all healthy infants? Did you were there some with some underlying conditions, or what type of samples did you sequence? And did you then find other shifts with different underlying conditions? I was wondering.
Yeah, thanks for your question, Adrian. So we didn't necessarily analyze the data ourselves in this publication. It's more of a perspective that we published presenting a possible framework that could explain the switch that's been shown. But in the papers that we're citing and what we're talking about, I think the majority of them were studying healthy infants, or at least at some point they had like a healthy control, and we were really focusing on healthy development in the paper. In terms of sampling bias, or just in general what the samples were, or how they identified, I guess, life cycle. For the lysogenic replication, that was through really just looking at bacterial lysogens and seeing that their prevalence seems to be much more common in the adult versus in the child. And also, trying to remember, for the lytic interactions in early life, there's some data showing kill the winner dynamics between phages and their bacterial host, which is essentially a type of coevolutionary dynamic where the most abundant bacteria are kind of predated by the most abundant phage, and this leads to cyclical shifts in species abundances, which isn't something we see as frequently in the adult gut. So the evidence, I would say, is still kind of lighter on that end, and it's definitely something we're exploring in healthy and also in malnourished children to see how strong it is and if our framework actually holds true. I think the phage directory email was just sent out, so there is a link in there if you want to, or for anyone, if you want to take a look in more detail at the framework.
I was wondering if you were going to use that high C method, how could you tell the difference between lytic and versus lysogenic? Or would that be just to tell the host?
Yeah, so I think high C really is like mostly for host. What's nice is they also are able to, or we're also able to resolve with that method co-infections by looking at how many different phage genomes are in or are associated with any given bacterial host. So with that, we can get a sense of co-infections, and that's something we're also proposing in the framework. But in terms of life cycle, probably we'd use like a different method to go about that, whether it's prediction tools that are out there, for example, phage.ai, or just looking at prophage prediction in the bacterial genome, read mapping to see if like free virus reads map to a bacterial genome, because that could be also an indicator of like an induced prophage, perhaps. So high C would be really more for the phage host kind of determinations.
I have a maybe stupid question, but also maybe a clarification point. What is the difference between the two models that you propose, which is that there's a kill the winner dynamics, and then there's also this arms race dynamics. Is there any difference between those types of dynamics in terms of bacteria phage interactions in the gut?
Yeah, so I'll just quickly summarize the dynamics for people on the call. Like I mentioned in response to a question earlier, kill the winner dynamics are probably really one of the best described kind of dynamics between predators and preys, where the most abundant bacteria are predated by kind of the most abundant lytic phages, and then this leads to kind of cycles of abundances up and down as lysis occurs. Arms race and fluctuating selection dynamics are more kind of discrete in a sense where it's really looking at how these species, how the phage and bacteria co-evolve. And for instance, bacteria may acquire a CRISPR spacer to some phage DNA sequence, but this would consequently select for different phages in the community that don't have that sequence, or that have like a SNP at that locus. So there, I think, and what we kind of mentioned in the paper is that it's probably it's not just one of these occurring in the gut at any time, but rather probably a bunch of different dynamics occurring between different phages and hosts over time. And we really stress the importance for longitudinal sampling, not just kind of snapshot or cross-sectional studies, to get a better sense of coevolution over time. And to look at that in more detail, we could look at not only DNA sequences of phages and bacteria, but also, for example, transcriptomes to see if bacterial gene expression is changing over time. That would maybe indicate, like if we see, for example, some membrane receptors are not as frequently expressed anymore, this altered, let's say, a phage abundance, perhaps that's some sort of arms race dynamics at play, things like that. So that's something that unfortunately hasn't been done to date. So there's not a lot of longitudinal infant, or really longitudinal gut virome and bacterium data out there. But I think with that data, it'd be really powerful to get a sense of the ecological dynamics there.
So just a point of clarification, and maybe this is because I'm misunderstanding the maybe is it a time scale thing? I don't know. I'm thinking about the two types of dynamics that you just exposed. So the kill the winner dynamics do not exclude an arms race happening between the phage and bacteria. It's just that the time scale is different, where you have more killing of, or more rapid killing. Is that something? Am I correct? So where's the difference? I'm sorry.
Yeah, I think so. At least that's my interpretation of these different dynamics. And also, kill the winner has really been described as more of just a cyclical pattern. Looking at phages and bacteria, you'll see kind of a nice curved sine wave kind of pattern in their abundances, whereas arms race dynamics and fluctuating selection, you may not see these kind of fluctuations in abundances if you're looking at their abundances, but rather you'll see changes in, let's say genomes or protein expression, things like that, or bacterial defense systems. And yeah, they do kind of go hand in hand, because there's always going to be that arms race between phage and bacteria as one becomes resistant and one overcomes resistance. I hope that made sense.
Yeah, it did. Thanks for that. That makes sense. Now, I have a question that might be a point of contention, which is, how much does CRISPR/anti-CRISPR play a role in the establishment of the virome communities in the infant gut? Is there something known about this?
It's a good question. I'm actually not sure as to like the prevalence of CRISPR systems in infants specifically. I know kind of just in general, like if we're looking at all bacteria, I think it is, like you said, a bit of a point of contention, but most people say between 10 and 40% of all bacteria have at least some sort of CRISPR system, although some are leaning down more towards the 40%, I think. So if that is kind of the same ratio in the gut, where let's say 1 in every 2 or 1 in every 4 bacteria has some sort of CRISPR system, I think, yeah, that would be a very important kind of defense system to study. And there's also a bunch of other phage defense systems which are being uncovered every day, it seems. So yeah, I think that's something that should also be looked at further and to see does the prevalence of these phage defense systems maybe increase as the child becomes an adult and as their community stabilizes, and making these bacteria perhaps more resistant once again to like phage infection or to community remodeling.
Thank you for answering this question in a more broad sense, which I was hoping to continue. Sabrina, you were going to ask a question. Sorry.
Yeah, I guess I have one more question. So I was thinking about phage hunting. Like, if you were going to try to find lytic phages, would you, to help patients, would you get a lot of it from infant poop? Like, should we be getting that source to find these lytic phages?
I mean, you could sell, I don't know if you know this, but you could sell your baby's meconium for a bunch of money. So maybe there.
I'll take anyone's.
How interesting. Okay. I'm not sure who the target audience would be for that, but
I have no idea either.
Yeah, good question. So I think, the communities that we see in terms of like viruses and bacteria in the infant are very different than what we see in healthy adults. So I think by adding, let's say, to your repertoire of kind of samples you're screening for phage, if you're adding these infant samples, you automatically have like a whole different set of phages and bacteria that you wouldn't necessarily find in a healthy adult, or really mostly any adult. So yeah, that could be a good way to kind of expand one's repertoire of phage. I kind of guess though, if people are looking at like sewage, that's a good mix of everything, so you may capture some of them in there anyway.
But it would be in a diaper, right? So it wouldn't go in the sewage. So I don't know.
Yeah, at least for a couple of years. Yeah, so you're right. It might just be missed completely for most.
Sabrina, you missed this opportunity with your kid.
Oh no, I did look.
Poop from diapers.
I did look.
See?
Okay. Did you find anything?
Well, I was looking for a particular type of phage, but no, I didn't. Okay. But I'm sure there are phages.
Yes, for sure. And I think when trying to isolate like phages for gut commensal hosts in general, I think a lot of microbiology being done like for phage isolation is aerobic. And like Jean-Paul mentioned, a lot of the gut bacteria are microaerobic or anaerobic. So we should really explore, I think, phage hunting in a more anaerobic environment as well, because I think there's a lot of diversity to uncover there.
True that. Okay, we're at the top of the hour. Does anybody have any more questions before we wrap up this Phage Friday? We didn't talk about printing, but we could print phages for the gut. Are there any more questions from the audience? Questions from up here? Maya, thank you for coming up and answering my questions about cocktails and use of cocktails in Georgia. And thank you so much. Are there any other questions from the audience before we wrap up? It seems there are not. So, Michael, thank you so much for coming. Thank you so much for accepting the invitation. Thank you so much for taking the time to talk about phages and babies and more for me cuties and more baby. I know, terrible jokes. I have terrible jokes.
Yeah, no, I saw the for me cuties. I think it was in Sabrina's.
That's my terrible joke. I have.
That's great. And also, I thought of Microviridae, right? The phage family. Micro babies are small. I don't know. Another bad time.
Baby Viridae, yeah, terrible. Other than that, it was awesome seeing you guys on Phage Friday. We'll see you next Phage Friday to talk about synthetic phages, right?
Yes.
We're talking about synthetic phages next Phage Friday. people, it was awesome talking to you on Clubhouse. I hope to talk to you not on Clubhouse or maybe on Clubhouse next Friday, next Phage Friday. Meanwhile, I will see you next Phage Friday.
Bye, everyone.
Awesome. Thanks again. And this was really fun. Bye, everyone.
Thank you.
Yeah, thanks.
Thank you, Michael.
Bye.